The production of recombinant proteins, especially membrane proteins, for structural and functional studies still technically challenging due to low level of expression and the inherent instability of many membrane proteins once solubilized in detergents. In response, many laboratories have implemented protein production pipelines to accelerate the expression and purification process. A common feature of these pipelines is the use of expression screening to quickly identify the conditions that will allow sufficient protein production on scaling up. One of the most used expression hosts is E. coli, and screening procedures are mainly based on decreasing culture volume and increasing parallelization, making it possible to work with 96 cultures or more simultaneously. This process enables more proteins or protein domains to be tested and several expression condition parameters to be optimized. One commonly used approach is to fuse the protein to green fluorescent proteins or GFP, enabling expression to be tracked without the need to purify the protein. In this way, information about expression level, stability, and behavior can be assessed with small amount of unpurified material. In this video, we will describe a streamlined protocol for expression screening of membrane protein in E. coli using fusion to GFP as a reporter of production. We are going to express four major facilitator superfamily drug efflux transporter from different Mycobacterium species in different E. coli strains and different autoinduction media. The genes have a GFP histag in each C-terminal for easy detection. For this experiment, to aid interpretation of the result, we include a positive control, a popping E3CE GFP, a plasmid that expressing GFP. Prior to performing this experiment, E. coli competent cells are made, aliquoted, and stored at -80 degrees Celsius. Tau competent cells on ice. Add 3 microliter of expression plasmid to each aliquot of competent cell. Incubate the mixtures on ice for 30 minutes. Heat shock the cells for 45 seconds at 42 degrees Celsius. Allow the cells to recover on ice for 2 minutes. Add 300 microliter of SOC media into 96 well plate. Add cells into the plates containing SOC media. Seal the plate and shake at 37 degrees Celsius incubator for 45 minutes. Transfer 30 microliter of cells from the transformation mixture into LB agar plates. Spread, dry the plate, and incubate at 37 degrees Celsius incubator overnight. Prepare 50 ml LB non-induced media. Then prepare 15 ml LB, SB, and TB auto-induced media. Inoculate 3 ml samples of LB non-induced media plus ampicillin. Seal with a breathable sealing. Incubate at 37 degrees Celsius overnight. This is the pre-culture for the expression screening. The next day, add 15 microliter of overnight culture into 3 ml XB auto media plus ampicillin in a 24 well plate. Seal with a breathable sealing. Shake the culture at 30 degrees Celsius for 24 hours at 200 rpm. After 24 hours, take out the cultures from the shaker incubator. Take out 1 ml of culture from the 24 well plates and transfer to a new 96 deep well plate. Pellet the cells by centrifugation at 2000 G for 15 minutes at 4 degrees Celsius. Remove the media by inverting the block and decant the media. Tap the block gently onto a paper towel to drain remaining media. Seal the plates and store at -80 degrees Celsius for a minimum of 20 minutes. Defrost the pellets in the 96 deep well plates for 20 minutes at room temperature. Place on ice and resuspend the cells in 200 microliter cold lysis buffer by pipetting up and down using a multi-channel pipette. Add 20 microliter of the LPI solution. Incubate for 20 minutes with shaking at 400 rpm at 4 degrees Celsius. Add 10 microliter of 25% DDM. Incubate for 20 minutes with shaking at 400 rpm at 4 degrees Celsius. Transfer the mixture into microtubes and centrifuge at 6000 G for 30 minutes at 4 degrees Celsius. Transfer 16 microliter of the cleared lysis and add 4 microliter of 5x SDS loading dye. Do not boil the samples. Before loading the samples, run the gel for 10 minutes at 120 volt to get SDS into the gel. Load 15 microliter of the samples, protein standard, and GFP control into SDS patch gel. And run at 150 volt for about 1 hour. Place the gel onto a BioRad imager. Set up the exposure time to ensure that the brightest bands are not saturated.